In this lecture, now that we've talked about how cells can transmit electrical signals and activate or excite a cell, we're going to link that excitation to muscle contraction. We'll start out by reviewing electrical excitable cells and the process of action potential, and then we will talk about how we get that signal from a neuron into the muscle fiber across the neuromuscular junction and ultimately link that excitation of a muscle fiber into a contraction. So electrically excitable cells are ones where the plasma membrane undergo a voltage change in response to stimulation. Most famously, or most well known about this, are the neurons, which convert electrical signals into transmissions where one neuron can send an excitable transmission that may change to yellow, send an excitable transmission over and excite another neuron, which can then send a signal over and excite another neuron. But what we're going to be talking about for the next couple lectures is muscle fibers and how they can be electrically excited. I like this picture. This is an electron microscopy image of a muscle fiber. Here you can see the myofibrils, and then this is the endomysium surrounding that individual muscle fiber. Electrical activity on cells is dependent upon a difference in ion concentration across the plasma membrane. And so here you can see extracellular fluid. We have a net positive charge, and relative to the extracellular fluid, there's a negative charge on the inside of the cell. In terms of where these charges come from, here you can see there's more positive on the outside, which is primarily dependent on sodium, and less positive charge on the inside because there's fewer potassium ions on the inside. So here you can see 1, 2, 3, 4, 5, 6, 7 times positive versus only 4 times positive. And really it's this difference between the number of positive ions on the outside versus positive ions on the inside that cause this difference in ion concentration. This membrane that we're looking at is polarized, and so we're going to be talking about membrane polarity a lot. So polarized, this is equivalent to how batteries are polarized as well. So you can see this is polarized because there's a positive charge on the outside and a negative charge on the inside of the membrane. The difference in electrical charge from one point on the cell to another, or one point in anything to another point. So if you're looking from here, you're looking at voltage versus here, is called the electrical potential. And electrical potential is measured as voltage. When a neuron or a muscle fiber becomes excited, this is caused by a rapid motion of or flow of sodium ions across the plasma membrane. Rapid flow of sodium ions across the membrane. So how does this movement change the polarity of the plasma membrane? When neurons or muscle fibers are excited and sodium ions flow rapidly across the plasma membrane, they become briefly depolarized. And so depolarized means that there's less difference between the outside and the inside. And so since the plasma membrane typically is negatively charged, less difference means that the difference is going to become less negative, or closer to 0 millivolts across the membrane. So when a membrane becomes depolarized, like I said on the last slide, there's no electrical charge difference. So this membrane, because there's no charge drawn in here, is depolarized. The plasma membrane then repolarizes as potassium ions. So potassium ions flow rapidly out. And they flow out through their own voltage-gated potassium channels. So you can see here potassium flowing back out, repolarizing the membrane and reinstituting that voltage difference. The rapid fluctuations in membrane potential that we see during cell signaling, which you can see from this point here up until basically we get to this point here, is called an action potential. Because as we talked about in the last time, this difference here is caused by a graded potential. And we'll talk about what happens if we don't hit threshold during a graded potential when we get to talking about neurons. To quickly review the events of inaction potential, starting with a resting state, here you can see we're at resting membrane potential. All of the membrane channels are closed, and resting membrane potential is maintained at roughly negative 70 millivolts. We then have some event that occurs that depolarizes the membrane. That brings us up to threshold right here. And so then the second step is depolarization, where the voltage-gated sodium channels open and the membrane depolarizes. Fairly quickly, we get to full depolarization. The voltage-gated sodium channels get closed by the inactivation gate. The voltage-gated potassium channels open, and the membrane repolarizes, going back to further from zero, so into the negative territory. It actually passes resting membrane potential and briefly hyperpolarizes as the voltage-gated potassium channels slowly close and the voltage-gated sodium channel resets itself, getting ready for another action potential. What we didn't talk about last time is how this signal actually turns into an event that can be propagated along the plasma membrane. In brief, propagation occurs as the depolarizing action potential of a neighboring region brings the next region over into threshold, activating the action potential. So here in this one, in section 1, you can see an approaching action potential coming along and depolarizing the membrane just in front of it. So if the action potential is right here, here you can see some depolarization reaching threshold. And so you get basically this wave of depolarization moving along the cell, going down one direction, so propagating in that direction. So in this point, the electrode is measuring one spot. Here we get depolarization, followed by a wave of, so here's the depolarization coming down, followed by a wave of repolarization passing down, and the signal transmits itself along the length of the plasma membrane. What is illustrated by this process is that repolarization is followed by a wave of depolarization along the plasma membrane. Having reviewed the action potential, we're now ready to talk about how do we excite a muscle fiber. And this excitation occurs at the neuromuscular junction. So if we look at all skeletal muscle fibers are innervated or activated by somatic motor neurons. Somatic, soma for body, motor neurons, which are the neurons that go to skeletal muscle. And motor neurons excite or activate all the skeletal muscle fibers. So anytime you hear the words motor neuron, you know that it is associated with a muscle fiber. The cell bodies for motor neurons are all located within the central nervous system, which includes the brain and primarily the spinal cord. And we'll talk about that anatomy as we get into the nervous system soon. So looking at a neuromuscular junction, there's a problem. The signal needs to be transmitted from the neuron, which here you can see in purple coming in, and then activate that associated muscle fiber. And so how do we link one cell to another? So and part of the problem is that motor neurons and muscle fibers are not electrically coupled. So you can't excite one and have a direct coupling to the next cell. So we need some way to jump that gap. So to jump across, we have this process of neurotransmitters. So neurotransmitters are chemical signals that carries a signal between the motor neuron and the muscle fiber. So we're going to talk briefly about how they do this, but it's basically a neuron coming in. You have a neurotransmitter that then comes across, and if this is skeletal muscle, is going to excite that skeletal muscle. We will classify types of connections between neurons and other cells in a little bit when we talk about neurophysiology, but just so that you can refer back to this, the neuromuscular junction, which is the point where a motor neuron excites a muscle fiber through these chemical signals, is called a chemical synapse. And so we're going to have a brief introduction to what is a chemical synapse. We will do it a little bit more in detail the second time we hear about them when we talk about neurophysiology. Anatomically, looking at a neuromuscular junction, there's only one neuromuscular junction per muscle fiber. So 1 NMJ per muscle fiber, which makes sense. So I talked about this earlier on when we first talked about the skeletal muscle system. You don't want more than one neuron controlling a muscle fiber, or else that muscle fiber will get conflicting information. So what one neuromuscular junction per muscle fiber means is that every time the motor neuron fires, so motor neuron which also means that it's excited, the muscle fiber that it's connected to will contract. And we'll get to how do we get various forces associated with muscle contraction in the next lecture. The process of muscle fiber contraction is broken into 4 steps. So excitation, so you excite the muscle fiber, you then have to couple that excitation to the process of contraction, so sort of linking excitation of a muscle fiber to the activation of contraction. We then go through the process of contraction with the cross-bridge cycle, and then ultimately we need to turn off contraction through relaxation. So the first step of muscle fiber contraction is excitation, and this is the event that occurs at the neuromuscular junction and is the activation of a muscle fiber by the motor neuron. So there are 5 steps that occur during excitation. First step is you need to have the signal or nerve impulse arrive at the nerve terminal. So in this diagram, here's the nerve coming in. So the signal is coming in, arriving at the nerve terminal, which is this blown out structure here. Second step is that nerve impulse needs to stimulate the release of acetylcholine from the nerve terminal. So acetylcholine released from nerve terminal. So it's these little tiny dots that are getting released into that synaptic cleft, which we will get into the anatomy of that later. Third step, acetylcholine diffuses across the synaptic cleft. So you can abbreviate acetylcholine ACH, diffuses across synaptic cleft. So 3 is this movement. Next thing, acetylcholine needs to bind its receptor on the surface of the muscle fiber and excite it, basically opening up sodium channels, creating the end plate potential. So ACH binds. Actually, we'll change that. So acetylcholine opens sodium channels on muscle fiber within the neuromuscular junction, causing the end plate potential, which is the muscle-specific version of a graded potential. So that will be happening down here. And then the fifth step of excitation is actually turning off the signal, in which case acetylcholine is then broken down and the signal is stopped. The second step of muscle fiber contraction is excitation-contraction coupling, and this is the link between muscle fiber excitation and muscle contraction. So here basically is a quick overview of what's happening, but we've got an action potential running across the plasma membrane, down the T-tubule, and then linking with the intracellular apparatus that then allows or activates contraction of the muscle fiber. So the first step of excitation-contraction coupling is the propagating action potential goes down the T-tubules. So action potential propagates throughout muscle fiber, including down the T-tubules. Step 2 is the release of calcium from the sarcoplasmic reticulum. So depolarization of the plasma membrane in the T-tubule causes calcium to be released from the sarcoplasmic reticulum. The sarcolemma causes sarcoplasmic reticulum to release calcium. Step 3 is the increase in intracellular calcium. So calcium in sarcoplasm binds troponin. Step 4, troponin moves tropomyosin off actins, myosin binding site. So then actin and myosin can interact. So in summary, here we have step 1. We have action potential propagates down the T-tubule. Within the T-tubule, the action potential depolarization of the sarcolemma causes the SR to open, releasing calcium, so calcium levels in the cell increase. Calcium in step 3 binds troponin. When calcium binds troponin, troponin then changes conformation, pulling tropomyosin off of the myosin binding site on actin, and then actin is exposed and we can get contraction. So that's step 4. So the next step that we need to talk about is how do we actually convert chemical energy, chemical potential energy, into mechanical energy. And so this is done through a process called the cross-bridge cycle, which we will talk through again in steps. Step 1 of the cross-bridge cycle is the cocking of the myosin head. So this is over on the left side here. Here you can see myosin in blue. And when ATP is hydrolyzed to ADP in phosphate, so this is ATP goes to ADP in phosphate, that energy that's being released from that hydrolysis causes the myosin head to go from its low energy state to the high energy state. So ATP goes to ADP plus PI plus work, and that work is applied to myosin head cocks, entering its higher energy state. So basically putting that potential energy from the ATP into the myosin head. Second step of the cross-bridge cycle is called cross-bridge formation, and it's this step where myosin binds actin. And at this point, as myosin binds actin, inorganic phosphate, so PI is inorganic phosphate, is released. So we've now, step 1 was putting myosin into its high energy state. Step 2 is sticking myosin to actin. Step 3, the power stroke, is where the thin filaments slide over the thick filaments. This thin filament, which again is actin, pulled over thick, which is myosin. And doing this is the release of myosin from its high energy state to its low energy state. Goes to myosin low energy. And as it does this, ADP is released. So adenosine diphosphate released. The last step of the cross-bridge cycle is cross-bridge detachment. And this is where we are now sitting here, sort of in this in-between stage. And detachment occurs as a new ATP binds myosin, breaking the actin-myosin bond. The last step of muscle fiber contraction is relaxation, where we stop or terminate the contraction. So first thing that happens is you need to stop exciting the muscle fiber, and so the nervous signal stops. So because of that, there's no more acetylcholine being released at the neuromuscular junction. Second step is acetylcholine needs to be broken down, and it is degraded by an enzyme called acetylcholinesterase. Acetylcholinesterase. So in this picture, the little Pac-Man thing, the dark purple, is acetylcholinesterase. Acetylcholinesterase. And the green dots are the acetylcholine. So you can see the acetylcholinesterase is coming in, breaking it down so that it's no longer available to signal the muscle fiber. What this little black arrow is showing is that the cells actually are very conservation minded and will recycle those components back into the neuron to make more acetylcholine. So once the acetylcholine signaling is stopped, the action potential stops, so you no longer have the excitation of the muscle fiber, and so then without a depolarizing sarcolemma, calcium gets reabsorbed into the sarcoplasmic reticulum. So 3 sarcoplasmic reticulum resorbs calcium. And this is actually just, there's a calcium pump that's constantly pumping calcium back into the sarcoplasmic reticulum. It doesn't actually turn off, but because the calcium leak channels have stopped being open, pumping calcium back into the sarcoplasmic reticulum actually moves it out of the sarcoplasm. Without calcium in the sarcoplasm, you then have troponin no longer attached to calcium, and so then tropomyosin can then block the myosin binding site of actin. Troponin dissociates from calcium and the fifth step is tropomyosin blocks myosin binding site on actin. So then the contraction stops. Just as another thing to see what you got from the lecture so far, what stage of contraction would muscles be in if no ATP were available, and when does the body run out of ATP? So when would this actually happen? So to think about this, so pause if you want to think about it for a second. So basically the step where the contraction, if you ran out of ATP, muscles, actin and myosin fibers would be stuck together. So it would be during the cross-bridge cycle. So actin and myosin bound. So, and the only point during our life or lack of life when this would happen is when we're dead. And so this is what is known as rigor mortis. So if you've seen a dead animal, or usually it takes 20 minutes or so after it dies, it's used up all its ATP, its actin and myosin cross-bridge becomes completely linked, and the animal goes into rigor mortis where it's very stiff. And then another 8 hours later, it will actually leave rigor mortis as bacteria start to break down the proteins, and the muscles can then relax again. So just to review really quickly, termination occurs as nervous signal stops, acetylcholine is no longer being released, it then gets broken down by acetylcholinesterase. Without the excited muscle fiber, calcium gets reabsorbed into the sarcoplasmic reticulum. With lower calcium levels, calcium detaches from troponin, and then without troponin pulling tropomyosin off of actin, tropomyosin falls back into place, blocking the myosin binding site and stopping the actin-myosin interaction.